Welcome to your zoology lab. I'm Dr. Jen Peak. I will provide most of your notes as videos. You will also have some additional notes that you'll need to complete. First thing is to make sure that you log into Moodle and you check out the notes that are available. Each lab is broken down into a different tab for you. So let's go ahead and get started on the integumentary system. I'm looking at page 7 of the lab manual and then the purple skin model that goes along with it. Alright, so hopefully you have the fill-in-the-blank notes available. As you go through this video, you will see that those blanks will be answered for you. And this way you can just kind of fill them in as we go along. Now, the model on the right is the actual skin model that we have in lab, and it's a three-dimensional model. And you can't really tell from this picture, but it comes out of the purple background, and there's texture to it. And so I'll go through these structures with you as we go through the notes. All right? So your largest organ that you have is your skin. Your skin is going to make up about 15% of your body weight. Now, it's broken down into two main layers. You have the epidermis, and the epidermis is going to be represented by numbers 1 through 5 here. Those are made up of stratified squamous epithelium. They also have some cuboidal cells and columnar cells on that bottom layer there. And I'll talk about those more in a little bit. But you have this epidermal layer, and then you have this deeper dermis layer. All right, and so that deeper dermis layer is identified at number 6. So the dermis layer is connective tissue, and then below it at number 7 is the hypodermis. Hypo tells me that it's low or beneath. So when I think about things that are hypo whatever, I typically think about like if somebody's hypoglycemic. That means they have low blood sugar. Versus hyper, which would mean that something's high. So if someone suffers from hypertension, they would have high blood pressure. So hypo tells me it's low or beneath, and it's low or beneath number 6, which is the dermis. So number 7 is your hypodermis. It's also known as fat or adipose tissue. And so again, it's another connective tissue, but that's an associated layer. So it's not going to be classified as a true layer of the skin like the epidermis and the dermis would be. Alright, so overall it's about 1 to 2 millimeters thick. The thinnest is at 0.5 millimeters. That would be at your eyelids. And then the thickest is between your shoulder blades at 6 millimeters. And also the palms of your hands and the soles of your feet are very thick. So let's talk about the function of skin. There are many different functions of skin. You'll notice that you have to have a certain percentage of your skin intact and functioning properly to sustain life. So first off, it is your first line of defense against infection and trauma. It's going to help repel things from entering the body. It's resistant to abrasion and penetration, and so it's basically preventing things from getting to that underlining tissue. Your skin is going to recover better than other organs. So that's one of the things about epithelial cells, that they recover very quickly when trauma does happen to them. So like when you think about like your liver regenerating, that typically happens fairly quick. Your eyes, damage to like the outer surface of the eye happens fairly quick. And then your skin is able to heal very quickly. And these are just common things with a lot of the epithelial tissue, but your skin is just really good at it. Now, there are also some things that are going to occur that help keep bacteria and fungus from growing on the skin. And those things are like your skin having antimicrobial properties, and then whenever you produce sweat, you also find that the skin is slightly acidic. So both of those are going to help keep bacteria and fungus in check. That's why you're hopefully not growing mushrooms underneath your armpits. All right. when that homeostasis is, that equilibrium is out of balance, that can happen, like say you're eating too many sugary foods and you end up more prone to like yeast infections, or hygiene isn't as good as it should be, and maybe you're ending up wearing like the same wet socks and shoes. You may end up with athlete's foot, or sports, for example, where an individual ends up with jock itch, right? So that can happen, but a lot of times there's some kind of environmental reason for it, or there's a health condition for it, right? And so that's going to influence the skin with respect to growing bacteria or fungus. So the other function is protection from sun rays. Your skin is basically going to be a barrier there to prevent the underlying tissue from being damaged and from sun rays or anything else. It also is going to prevent the absorption of water. So what you'll find is that the outer layers of the epidermis, so like number one, especially number one, it's very flat keratinized cells. They are scaly. They end up being very resistant to abrasion and penetration. They will eventually flake off. So what essentially happens is you start off at number 5, and then over the course of about 3 weeks, those cells push up, and they go from 5, 4, 3, 2, 1, and then flake off. So as they push up, they begin to produce keratin filaments. Those keratin filaments cause the cells to become flat and hard, and then they flake off. But it allows that outer surface to become more structurally rigid and able to prevent penetration. And so that's going to help also repel water. And this is why you're not like SpongeBob SquarePants and you just absorb all this water. Those outer layers there are going to help repel it. In addition to that, between number 3 and number 4, you actually have an epidermal water barrier. This epidermal water barrier will have embedded proteins that are within lipids, and so it's going to repel the water as well. And so water that's on the inside should stay on the inside, and water on the outside should stay on the outside. And then the only way we're going to get water from the inside out is through these structures like number 21 and 23, which is the longitudinal section of the sweat gland. Those structures there can actually pull sweat from the glomulus and move it up to the skin, and so we can get water out that way. But otherwise, it's going to prevent the absorption of water. Now, the other reason that you have to have skin is to actually start synthesis of a specific vitamin. That vitamin happens to be vitamin D. So to have vitamin D synthesized, you have to have a certain amount of sunlight every week. Now, the literature varies on how much sunlight is needed. Some say that you need several hours, like 15 hours or more. Others say that you only need like 6 hours, or that, enough indirect sunlight or whatever. Some literature states that there's a correlation between decreased vitamin D levels and obesity. Other literature says that that correlation is more so due to individuals who are obese not getting outside enough, and when they're not getting outside enough, they're not getting enough exercise, and so it may not be that the decreased vitamin D actually causes obesity, but that they're not getting enough activity, and that's why we see this correlation. So, but anyway, you need to have sunlight in order to synthesize vitamin D. Specifically, we're thinking like D3, right? So how does that work? Well, the way that works is that vitamin D3 has to have another molecule actually help break it down. And that molecule doesn't get activated unless you have a certain amount of sunlight. So basically think of it as this molecule has to have an alarm clock. And the alarm clock for this molecule is the sun. So when you get enough sun, it activates this precursor that will go and break down vitamin D. If you don't activate the precursor because you don't get enough sun, then vitamin D doesn't get synthesized. It doesn't get broken down, right? There's no catabolic reaction that happens there. All right, so you have to have sunlight. It has to activate the precursor. The precursor then can go find vitamin D and break it down. So let's say I'm some kind of crazy person and I abduct you and I take you home and I store you in my closet. You're not getting any sunlight in that closet, right? So whatever vitamin D you have in your system, you can't utilize because it cannot be broken down because the precursor isn't getting activated if you're being stored in a closet. Now, if I were to try to pump you full of vitamin D, like feed you a lot of, like you drink nothing but milk, good enhanced vitamin D milk, you still cannot process all of that vitamin D because you're not activating the precursor in order to break down the vitamin D. Now, you're going to find that your skin is your most extensive sense organ. What does that mean? It means that your skin has a ton of different types of receptors that identify sensory information. All right, let's look at these. So you have, let's start off here at number 28. So 28 is kind of in the middle of your screen. It's right underneath that pink wavy line there. It looks kind of like a jalapeno pepper. And 28 is known as Meissner's corpuscle. Meissner's corpuscle is highly concentrated in your fingertips, your lips, your face, your neck, your genitals, the nipples. That's where you have a lot of Meissner corpuscles. Meissner corpuscles, if you think about those areas of your body, they are very sensitive to touch, specifically light touch. You can feel like if somebody were to barely touch you there, you would feel it. And that's because Meissner's detects that. So Meissner's corpuscle is there at 28. And if you go over to the right, you're going to see that there are, it's a little white looking couple lines that come up, and they're actually going to form these little balls at number 30. So again, we're right underneath that pink wave. We're on the right hand side of the model, and number 30 is kind of in a valley on the wave, and then it's pointing to these little white balls that then have a white streamer line that comes down. So these are known as Merkel's disc. Merkel's disc is also going to end up being for touch, light touch. If you look at your answer key in the lab manual, it's going to tell you that it's tactile, right? So it's for light touch. Next to this, keep going to the right, there is another white ball that ends up coming down with some streamers. And this one, it kind of looks like a balloon. This one is known as the bulb of Krauss. Now, this is one that typically when we are in face-to-face lecture, I'm going to tell you not to worry about this one, that you can remove it from your list. Again, it's tactile, right? But the bulb of Krauss, it's misleading. You have sweat glands here. It's often found with mucus glands, so you don't have mucus glands and sweat glands together. So this one is one that I usually tell you, just go ahead and mark it off your list, number 32. All right, so let's go back to Meissner's and Merkel's. All right, so Meissner corpuscle, Merkel's disc. You'll notice that they're both about the same level, all right, the same depth on the skin. They're right there by that pink wavy line, all right? That's at your stratum basal or stratum germativium. So this here, because they're at about the same level, they should have the same function. They're going to detect light touch, right? Not evenly distributed throughout the whole body, right? You need them highly concentrated in places that are going to be, especially Meissner corpuscle, highly concentrated in places that are more sensitive. Now, from this one, go down a little bit deeper, right? You have number 31 underneath Meissner corpuscle at the very bottom of the dermal region. You see number 31, right? 31 looks like a flat mushroom, right? You're spotting it. 31 is Ruffini's corpuscle. Ruffini's corpuscle is for pressure, as well as number 33, right? 33 looks kind of like a chili pepper, another jalapeno. I think pepper with this one, and it's known as your Pacinian corpuscle. So Ruffini and Pacinian are both at a similar depth on the skin. They are both going to deal with pressure. So your one that looks kind of like a mushroom, and then your bigger pepper over here, both deal with pressure. How I remember these: Pacinian looks like a pepper, so I think the P and the P. And then Ruffini corpuscle, the way I remember Ruffini is that Ruffini seems, years ago there was like this Italian restaurant and it was named Ruffini, and anyway, that's how I remember it, because I think about mushrooms on a pizza, and so that's Ruffini's corpuscle. So you have these here that are going to help with pressure and touch, and then look at number 29 in the middle of your hot pink area of the epidermis. So it looks like a tree, right? You see it? This is known as free nerve endings. Your free nerve endings are going to detect pain and temperature. And it makes sense that if you have something that detects pain and temperature, they need to be closer to the surface of the skin. If they were deeper down and you have, it needs to register that your hand is about to burn up, it would take forever for it way down here at the bottom near, Pacinian corpuscle for that to be detected. It needs to be closer to the surface so that that reaction can happen faster and you pull your hand away. Now, in addition to all of these, you're going to have a couple other things that will help out with being a very extensive sense organ. If you look, you have number 20 here. 20 attaches to the hair. It's one of them that's going to erect the hair. It's known as your erector pili muscle or pilio-erector muscle. So that's one that causes the hair to stand up. Typically when your hair stands up, it gives you a signal that it's cold or that there's fear involved. And your erector pili muscle is also known as a vestigial structure. So vestigial structures are those that we're not really sure why they're here. It might have been something related to our ancestors, and it has stuck around though. And so with this one, it generates a little bit of heat when you get cold. It allows the hair to stand up, so it allows us to know that something's wrong, but yet we might not really need help with hair standing up. All right, so there's a reason that we have all of these. And why do we have all of these different sense organs? Well, the purpose for that is for protection. Right? So that blank underneath most extensive sense organ, it's for protection. That's why you have that. Your next big blank is going to be thermoregulation. Thermo telling me temperature, regulation telling me obviously that it regulates the temperature. And there's many structures on this model that will help your body regulate your temperature. So let's discuss those. Sweat glands. We mentioned the sweat glands before. So here's the deal with your sweat glands. Your sweat glands are at number 21, and then a longitudinal section of them is at 23. When it says longitudinal section, it basically means that we shaved it off, right? So we cut it long ways. So you have 21 and 23. There is a glomulus of the sweat gland at 22 and 24. 24 is a longitudinal section again. You see that? All right, so you have sweat. Sweat obviously helps you cool off a couple ways. One, it's taking heat within that liquid, that sweat, and it's pulling it to the surface of the skin. So you have heat leaving the body that way. And then once it's on the skin surface, water or sweat is on the skin surface, you have evaporative cooling. Evaporative cooling is basically going to allow the heat to diffuse through the skin quicker. So the way evaporative cooling works is kind of like if you were to take a shower, right? When you get in the shower, if the room is, say, 72 degrees, it may not feel very cold. But when you get out of the shower, the room often feels colder than 72 degrees, and yet if you were to look at the thermostat, you're probably still at 72 degrees or close to it. But it always feels a whole lot cooler than that, and that's because of evaporative cooling. Your body is losing heat. Another thing that happens are these blood vessels. So your blood vessels go through what is known as vasoconstriction and vasodilation. They get bigger and smaller. If we were to go run laps, and every time you made it around the lap, I would take your temperature on your forehead, we would find that as you continue to run, your forehead temperature gets higher and higher and higher, and your face becomes more and more red. That's because the heat is coming to the skin surface so that it can diffuse through. But at a certain point, that temperature starts to drop on the skin surface. And you would think, oh, well, I must be sweating. Well, it actually drops before you even start sweating. And that's because the blood is needed at the muscles of the legs because you're running. And so the blood vessels to the skin get very small, and the blood vessels to the legs get larger. And with that blood goes the heat. So the skin temperature drops, and then your body has to figure out another way to decrease your body temperature. So you begin to sweat. Right? So sweating was one, and then vasoconstriction, vasodilation is another example. The hair is an insulator. So your hair itself would be another example. Your erector pili muscle causing goosebumps. When we have goosebumps, often it's a sign that we can apply friction, so start rubbing that area to get it to warm up, or cover up. And so that's going to also send a signal that tells us that. And then remember number 7, your hypodermis? That's fat, and fat's also an insulator. Right? So it's an energy storage site and insulator. So that's various ways that thermoregulation is going to happen with your body. Communication. Your skin is an excellent communicator. So it works with the muscular system, which then works with the skeletal system in order to give information, right? So facial expression and whatnot. Color of your skin can tell us information about health issues. So if somebody is red, it's possibly fever. They're overheated, there's an infection. If they're blue, that's cyanosis, which means that there's either a pulmonary or circulatory issue. There's a lack of oxygen. If they come in and they're jaundice, yellow, then that means that there's an increased amount of bilirubin in the blood, and so there's something going on with their liver. All right, so good communicator. You also have the storage of nutrients, and in this example, you have that hypodermis again, which is fat. So you have the storage of fat there. So let's talk about those layers of the epidermis. So there's several layers to the epidermis. You have 5 of them that are listed on this model. Not all 5 of them are throughout your entire body though. Okay, so as we progress, you're going to see this. Number 1 is going to be stratum corneum. Your stratum corneum layer, remember, is thick. It's a thicker layer. You can see here that it's thick, but it's keratinized cells. So they're dead, they're scaly. This is the layer that's going to be resistant to abrasion and penetration. It's also going to help prevent water loss. Layer number 2 is stratum lucidum or lucidum. This is going to be one that is only found in thick skin, so the palms of your hands, the soles of your feet. So it's a translucent layer here. It has no nuclei and no organelles. And this makes sense because we have pushed up past that water barrier and we are no longer receiving nutrients at this level. So there's no reason that any of these cells should have a nucleus or organelles because they've already started dying off and they're going to end up losing that. You have stratum granulosum at layer 3, right? So that's the one that has, the cells have become flat. They have keratinocytes. They're classified as keratinocytes, which means they have keratin filament or keratin. When you see big words, start breaking them down. So keratino tells me that there's keratin filament. That's the same thing that like makes your nails hard. And then cyte tells me that it's a cell. Okay, so don't freak out with these bigger words. Break them down. Remember that between number 3 and number 4, you have your epidermal water barrier. So epidermal water barrier with lipids and proteins. That's what we're going to find here. Think about what lipids are. Lipids are waxes, they're fats, they're oils, right? They're hydrophobic molecules. They're going to repel that water. So water on the outside stays on the outside between those flat keratinocytes and then the epidermal water barrier. And water on the inside is going to try to stay on the inside unless we allow it to come through with sweat. You have stratum spinosum or spinosum. You have 7 layers there of those keratinocytes. The ones that are deeper down actually can make new cells. And so what do we call it when one body cell makes another body cell? It's known as mitosis. Remember mitosis. Remember meiosis or meiosis would end up being making sex cells, eggs and sperm. Mitosis, remember when you go through prophase, metaphase, anaphase, telophase, that's going to end up being what makes your body cells, so making more skin cells. So those deeper layers here at layer 4 are able to reproduce through mitosis. As those begin to push up, they're going to stop dividing, so they're no longer able to do mitosis. They're going to end up producing more of those keratin filaments. And what do those keratin filaments cause the cell to do? They cause it to become flat, and eventually it's going to become hard. All right, so it's going to allow the cell to become flat. Number 5 on your list, that very last one there, this is your basement layer. I like to think of it as a basement layer because it's stratum basal or bacilli. In your lab manual and in some other texts, they can also call it stratum germativium. Right? Either one works. This is going to be a single layer. It has cuboidal or low columnar stem cells. Remember, cuboidal are kind of like those cube-shaped cells that you find in the kidneys. It's still epithelial. Columnar cells were more column-shaped. Those were found in your intestines. So if you think back to like that freshman biology lab, when you looked at histology, it might remind you of that. You're also going to have some keratinocytes here, but basically what you have is this resting bottom layer, and this bottom layer is known as your basement membrane. All right, so basilar, the basal, stratum basal, it's going to be on this bottom basement membrane. That last blank there should be basement. All right, so let's talk about the dermis as a whole here. So number 6, 0.2 millimeters at the eyelids, and then you're going to have 4 millimeters thick at the palms of the hands and the soles of the feet. So the reason it's thick at the palms of the hands, soles of the feet is that it gives you more cushioning in those areas. All right. So there's some other structures that we kind of hopped over real quick on here and didn't mention. So let's kind of go through a couple of them that we might have skipped over. We talked about the sensory nerves and the sensory receptors. All right, so those you should be good at. You'll notice that number 35 and 34 you cannot see on this model. And the reason you can't see it on this model is because it's 3-dimensional. So you're not able to see the side view of it. But number 1 through 5, right, if you were to look to the side along number 1 through 5, far left, you'll see that there's kind of sort of a number you can make out there, and that's number 34. That's your epidermis. And then your dermis is going to end up being along the side, closer to number 7, but on that far left side, and that's going to be number 35, the dermis. So the dermis could be labeled there, but more often than not, the dermis can be labeled at number 6. All right. All of your structures that are listed for the epidermis we have discussed. So numbers 1 through 5, the dermis at number 6, and then adipose, hypodermis, fatty tissue, all of those terms for number 7. So going through the accessory structures. So with the accessory structures, you have number 8 at the hair shaft. You have number 9, which was that longitudinal section of the hair. So longitudinal telling me that we cut it long ways. You have number 10, which is the sebaceous gland. Now, if I were to ask what comes out of this, what is secreted by it? Well, what comes out of a sebaceous gland? Sebaceous glands are named sebaceous gland because of sebum, right? S-E-B-U-M, sebum. But what does that translate over to? It means that it's oil. So where you see number 10 and number 11, that's going to be your commonly referred to as an oil gland. But you're not going to call it an oil gland in here. You're going to call it a sebaceous gland. All right. Number 11 shows you the longitudinal section of it. Number 12 is your hair bulb. 13 is your external root sheath. All right, so are you seeing that one? It's kind of in the middle bottom. All right, so we're looking at that external root sheath, number 13. In the middle of your picture, in the hypodermis region, going around that hair bulb there. All right, number 14 is the longitudinal section of the hair bulb. Number 15 is your internal root sheath. Number 16 is the growth and differentiation zone. So it's the part of the hair that has the matrix, the medulla, and the cortex. Number 17 is the papilla of the hair. You have veins and arteries at 18, 19. You also have them at 26 and 27. You have your erector pili muscle or pileo-erector muscle, which is your hair erector muscle at number 20. You have your sweat glands that we talked about at 21. Remember that 23 was the longitudinal section of those. You have the glomerulus of it at number 22 and the longitudinal section of that at number 24. You have the vascular papilla of the dermis at number 25. So if you're like, oh man, where's that at? Okay. Where you have number 23, the longitudinal section of the sweat gland, come down and it's those blood vessels right there next to it. Basically where the little artery and vein there connect. And then that's it. So the skin model should be a pretty simple one for you. Alright, so you definitely need to review this. Read over the part in your lab manual about the skin. From here, you need to start studying the information that's provided for you in the lab manual with respect to terminology and whatnot. So specifically, you're going to read page 5. Alright, and then you also need to go over page 2 and page 3. Page 2 will give you skeletal terminology that's going to help you out with the next class. Alright, so why is this process called such and such versus just being called a process or whatever? You need to read through that information. And then you have your cartilage and your bone pictures. So the histology information. So read over that stuff. Alright, I will talk to you guys on the next episode.